Hello and welcome to part 3 of the microbiology section for NBCE Part 1 board review. This is one of the public videos. If you want more content on this and an entire array of Quizlets, practice questions, full note sheets, as well as a whole bunch more lecture videos, in the description below there's a link that you can access a form to reach out to me, to contact me, to get the entire set of NBCE Part 1 review material. Thanks for watching and enjoy this video. All right, cells of the immune system. So we covered a good chunk of the most important ones in the clinical pathology portion. Just a couple more little details here that we'll pick through. So interleukins, these are chemicals produced by immune cells. There's three main ones that we find. Interleukin 1, its whole function is to activate lymphocytes. So it activates white blood cells. It's made by the macrophages, specifically for the brain and the nervous tissue. So the macrophages, they find something that's not supposed to be there. They latch onto it. They begin to break it down, and they release the interleukin 1 chemical to bring other immune cells to the area to help to fight that infection. Now, interleukin type 2 is T cell growth factor. If you're going to get asked a question about interleukins, interleukin 2 is the one that you're more likely to get asked about. And that's because it's made by the CD4 cells, also known as T helper cells. These are some of the cells that are responsible for directing and controlling the entire immune system. When we have specific conditions that attack the immune system, like HIV or AIDS, that is a condition where the CD4 cells are destroyed. So HIV destroys the CD4 cells, which is why it's such it can be such an aggressive virus, because it essentially destroys our entire immune system. So CD4 cells are connected to interleukin 2. Interleukin 3, it's made by the bone marrow, found in blood and lymphatic system. Nothing super specific that we absolutely need to remember about those. Major histocompatibility complex, known as MHC. There's two types, type 1 and type 2. These are going to they go on the surface of cells in order to help us identify what type of cells need to be attacked, to be broken down, because they are invading cells. They help us differentiate between what we say is self versus non-self. The main question that we get asked about with MHC is when we have tissue grafts or organ donation or organ transplant, and suddenly we have a different type of cells, a different type of tissue, different type of cell inside of the body, and the body can tag it as non-self and begin to attack it. Specifically, the major histocompatibility complex type 1 is going to work with T cells, and then both of them work with B cells. That's as deep as we really want to go into all of that. There's a lot more that we could get into, but boards is typically going to go more surface level with this kind of stuff and just memorize, what is the specific connection or the specific function of these things. Antigens, antibodies, and then something called complement. So antigens, these are substances that the body is going to recognize as foreign and can trigger our immune system. So very similar, like we have self versus non-self. We keep coming back to that kind of topic. The immune system is looking for anything that identifies something as non-self or invading. And antigens are these protein or polysaccharide components that are not found in our body naturally. They're found on top of other types of cell or other types of proteins that are not supposed to be in the body. We have a couple different types of antigens. We have complete antigens and we have haptens or partial antigens. These complete antigens, they have everything that they need in order to trigger an immune response all by themselves. Haptens, these are components of antigens, but they're too small to actually produce an immune response until they bind to another carrier molecule, another protein, another something else to give them enough form to actually trigger an immune response. You don't need to go too deep into all of this. The vast majority of this is just going to be word recognition. So we want to recognize what a hapten is, what a complete antigen is, by definition, to answer any questions about those specifically. A determinant, also known as an epitope, these are specific parts of the antigen that are recognized by the antibodies or our immune cell. So right here, this is going to be our antibody. This is the shape of the antibody. The antibodies have a constant region, which means that's all kind of the same. And then variable regions, these are built to recognize specific epitopes. So we have our antibodies or immune cells that have these variable portions. They recognize the epitope, and then they respond to it specifically. So this is why in all types of different reactions, especially like allergic reactions or response to antigens, the very first time we're exposed to one of these, the reaction might not be as severe as the second or the third time we're exposed to something. So, for example, let's say we're allergic to peanuts. The very first time we get exposed to peanuts, we might have a scratchy throat. And so we're allergic to peanuts. We might have a scratchy throat. We might have some hives. And it's uncomfortable, but it's not life-threatening. Then the second time we're exposed to the peanuts, that's when we go into anaphylactic shock. Our throat starts to swell up, all that kind of stuff, because the first reaction primes our body and produces this memory to recognize an epitope. And then the second time we're exposed to it, the body responds much more aggressively and much quicker to it. Additionally, kind of a secondary word is called an immunogen. Immunogen, these are always going to cause an immune response, whereas an antigen may or may not cause an immune response. Mostly just the focus on that is the immunogen, in case you get that word as a question. Antibodies themselves, these are proteins made by the B cells, specifically B cells that have converted to plasma cells. So this is where some of the questions will try to trick you. We're talking about plasma cells versus B cells. It gets a little confusing. So in order to produce an antibody, a B cell gets activated. When the B cell is activated, it converts and transforms into a plasma cell. Once it is a plasma cell, as a plasma cell, it then produces antibodies. So when you get a question, antibodies come from which of the following? Plasma cells is always going to be our best answer because it is the most direct link to antibodies. B cells is a good second answer choice. Within the antibodies, we have two general regions. We have an FAB region and the FC region. FAB, only connection that we potentially get asked about with this, the AB stands for antigen binding. This is the specific portion right in here where the antigen is going to bind to the antibody in order to produce the reaction that we want. Different types and classes of antibodies, also known as isotypes. We can use the mnemonic GAMED. We have IgG, A, M, E, and D. This is where a good chunk of questions tend to come from because each of these has a very specific reaction And so with pathology and with microbiology, we're going to get questions that give us information like blood draw was done, and this is the specific type of antibody that was found. So IgG, this is the most abundant antibody in the blood. One really cool thing about IgG is that it is one of the only antibodies to cross the placenta. So while still in utero, IgGs are the main antibodies that the mother is sharing with the baby, and this is important. Babies get their immunity from their mothers for the first about 6 months or so of their life. So in utero, IgG crosses the placenta, and this starts to produce our long-term immunity. IgG is also the first antibody to increase with that secondary exposure like I was talking about with that peanut example. Next, IgA. This is a secretory antibody, meaning that it tends to be found in the secretions of our bodies, saliva, tears, as well as the breast milk, and then also mucosal areas. A lot of the times they're going to give you at one of these two questions, which antibody do you get from the mother? IgG is our best answer. IgA is our second best because IgG crosses the placenta, and if the mother breastfeeds, they also get IgA from the mother. So if you get that question right here, which antibody do babies get from their mothers? Best choice IgG. Second best choice is IgA. When we talk about the condition multiple myeloma, we talk about the AG ratio reversal. This is what we're referring to. The number of IgA, because IgG should be the most abundant in the body. If we take the blood and it's actually IgA, that's a sign of multiple myeloma. Next, IgM. IgM is the first antibody to increase with our first exposure. It's the largest and the heaviest of the antibodies, and it has a pentamer shape, which means it has 10 binding sites. Those are all the most common questions that get asked on part 1. IgE. IgE as well, you almost always have at least 1 or 2 questions focusing on IgE. IgE shoots up due to allergic reaction, parasitic infection, or Hodgkin's disease. Those are the 3 specific things you're going to look for. If you have elevated IgE, either they're having an allergy to something, they got bit by some type of bug, most commonly a tick or a tick is one of the most common ones, or they have like a tapeworm or something else inside of their bodies, or they have Hodgkin's lymphoma. IgD, IgD really isn't fully understood, so you're not going to have questions about IgD. Antibodies mainly do 3 things. They do neutralization, so they bind onto toxins and viruses to stop them from going anywhere. They do opsonization. This is where they coat the pathogens in order to encourage phagocytosis. So they essentially coat them so that way they can't escape anywhere, and then they mark them and say, hey, this needs to be destroyed. This is a foreign invader. Complement activation as well. The classical pathway, meaning the pathway of inflammation and immune system and immune cell production, is initiated by the antibody complex, by an antigen antibody complex. So when the antigen binds to the antigen binding site on the antibody. Complement itself, this is a group of about 30 proteins whose job is to enhance or complement the immune system's ability to clear out microbes as well as damaged cells. The purpose is to enhance the phagocytosis. I'm not going to go too deep into all of this because there is like a whole cascade pathway that we could go through, but it's just not really worth the memorization component of it in order for boards for how many questions they typically ask about it. The things that we do want to remember is C3b, that's complement 3b, is where our different pathways, our immune pathways, meet each other and then continue. So our classical immune system pathway is triggered by the antigen antibody complexes. The alternative pathway is triggered directly by the pathogen itself. Those are the big things that we want to remember. The main functions of our complement, again, opsonization. They produce the inflammation, and also lysis. They want to break up pathogens and destroy them. Okay, and the next video we will talk about the hypersensitivities, which we did already cover in the clinical pathology, but we're going to go over them again here.